ध्याता ध्यानं नारायणः परः यच्च किञ्चित् जगत् सर्वं दृश्यते श्रूयतेऽपि वा अन्तर्बहिश्च तत्सर्वं व्याप्य नारायणस्थितः अनन्तमव्ययं कविगुं समुद्रेऽन्तम् विश्वशम्भुवम् पद्मकोशप्रतीकाशगुं हृदयं चाप्यधोमुखम् अथो निष्ट्यावितस्यान्ते नाभ्यामुपरि तिष्ठति ज्वालमालाकुलं भाति विश्वस्यायतनं महत् सन्ततगुं शिलाभिस्तुलं हत्याकोशसन्निभम् तस्यान्ते सुशिरगुं सूक्ष्मं तस्मिन् सर्वं प्रतिष्ठितम् तस्य मध्ये महानग्निर्विश्वाक्षिर्विश्वधोमुखः सोऽग्रभुग्विभजन् तिष्ठन्नाहारमजरः कविः तिर्यगोर्ध्वमदशायी रश्मयस्तस्य सन्तता सन्तापयति स्वं देहमापादतलमस्तकः तस्य मध्ये वह्निशिखा अणीयोर्ध्वा व्यवस्थितः नीलतो यदमध्यस्ताद्विद्युल्लेखे बभास्वरा नीवारशोकवर्तन्वी पीताभास्वत्यनूपमा तस्याः शिखाया मध्ये परमात्मा व्यवस्थितः स ब्रह्म स शिवः स हरिश्चेन्द्रः सोऽक्षरः परमस्वराट् ॐ शान्ति शान्ति शान्तिः।
संततगुं शिलाभिस्तु लम्बत्याकोशसन्निभं तस्यान्ते सुशिरगुं सूक्ष्मं तस्मिन् सर्वं प्रतिष्ठितम्. In this macrocosm of so many things, there lies a microcosm. Tasyante sushiragum sukshmam tasmin sarvam pratishthitam. That microcosm reflects in the macrocosm. Om Shri Sai Ram. Respected guests for the day, Professor V. Nagaraja and Professor D.N. Rao, HOD, Department of Biosciences, Professor S. Venkatesh, faculty, research scholars, and my dear brothers. A very loving Sai Ram to one and all, and welcome you all to the inaugural session of Gopinath Rao Memorial Lecture as part of International Microorganisms Day being celebrated by Department of Biosciences, Sri Sathya Institute of Higher Learning. As part of International Microorganisms Day celebrations, we had a hands-on workshop yesterday, and today we'll be having the Dr. K. Gopinath Rao Memorial Lecture. We'll start the proceedings by the ceremonial lamp lighting. I request the guests for today, Professor V. Nagaraja, Professor D.N. Rao sir, Director Dr. K. Nagunatha Sharma, Professor S. Venkatesh to join us on the dais for the ceremonial lamp lighting.
check, 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 check. Coming here.
Thank you, sirs. Firstly, I would like to request Professor S. Venkatesh, HOD, Department of Biosciences, Sri Sat Institute of Higher Learning, to deliver the introductory remarks for today's Dr. K. Gopinath Rao Memorial Lecture. Professor S. Venkatesh.
Hi Ram, respected Professor D.N. Rao sir, Professor Nagaraja sir, respected directors, HODs and faculties and dear students and research scholars. So today is the International Microorganisms or Microbiology Day. The thought brings with the invention of the microscope, we got a peep into the lives of microorganisms which otherwise were elusive to our normal eye. So this has opened up and revolutionized various aspects in human lives. There was a time when pandemics and other things had taken a big toll, as even evident from the recent COVID, which emphasizes the importance of understanding the micro world for our own existence. So before even the advent of man, the microorganisms were there, they have evolved, and with the advent of humans as one of the most dominant species, we have also played our role in their evolution. So one of the earliest way of selection and evolution were to use microorganisms for our own benefit, like production of wine or curd, etc. And then when we understand more about them regarding their role in diseases, though it is only a minuscule of microorganisms that are involved in such things, though they cause heavy losses to us. Over the years, again, new fields have opened up where we talk regarding how microorganisms in the gut modulate our own actions and how they are affected in various diseases. In addition, there is industrial growth where biotechnology has paved the way for using microorganisms to produce therapeutic proteins, diagnostics, etc. So currently, even there is no sphere in our life where microorganisms has not played a role. So it is good for us to have or celebrate this microorganisms day to understand the role of microbes and their uses for our own progress, social progress, and mitigating whatever problems are there in human lives. With this, I request Mr. Arun Sai to take over the stage for inviting Professor Nagaraj sir for the talk. Thank you so much.
Thank you so much, sir. Dear brothers and sisters, now I have the great privilege of introducing Dr. K. Gopinath Rao, in whose memory this lecture has been convened. Dr. K. Gopinath Rao was born on September 12, 1928, into a family of physicians. His father, Dr. K. Vasudeva Rao, was a well-known physician in Madras and the second superintendent of the famous Tambaram Tuberculosis Sanatorium, while his maternal grandfather, Dr. V. Srinivasa Rao, resident medical officer of a government general hospital in Madras, had the rare privilege of helping to care for Sri Ramana Maharshi in his later life. Dr. Gopinath Rao's parents prized education and service to others from a young age, and he observed his mother, Srimati Saroja Bai, and Dr. Vasudeva Rao, his father, as a model of inclusion and generosity in their large joint family. Dr. Gopinath Rao imbibed these quantities in great measure. He did his pre-university course at Loyola College, Madras, and then joined the Madras Medical College, graduating in 1953. His heart was set on becoming a surgeon, so Dr. Gopinath Rao trained at the Government General Hospital under Dr. U. Mohan Rao and at the Perambur Railway Hospital in Madras. After further training in England, he qualified as a fellow of the Royal College of Surgeons of England, FRCS, in 1961. Seeking to specialize further, he traveled to Canada, where he earned his fellowship in urology in 1968, and subsequently his second FRCS in Canada in 1970. Following his training, Dr. Gopinath Rao settled in Canada with his wife, Srimati Kala, and 3 children, where he practiced as a urologist, first in Brandon, then in Manitoba, then in Cornwall, Ontario, Canada. For more than 3 decades, he cared for countless patients with great skill and dedication. In addition, he was passionate about advising the field of urology and advancing it by publishing several articles in notable medical journals. In 1992, he was blessed by Bhagavan Sri Sathya Sai Baba to relocate to Puttaparthi to join the then nascent urology department in the Sri Sathya Sai Institute of Higher Medical Sciences in Puttaparthi. For his children and family, Dr. K. Gopinath Rao was a beacon of compassion, tolerance, patience, and humor. And for his colleagues and patients, a meticulous surgeon of great talent and deep humanity. For the inaugural edition of Dr. K. Gopinath Rao Memorial Lecture, we have Professor Valakunja Nagaraja. Professor V. Nagaraja received his BSc and MSc degrees in the year 1973 and 1975 from Bengaluru University. He then completed his PhD in 1981 from Department of Microbiology and Cell Biology at IISc on the role of DNA gyrase in mycobacteria. He then went on to pursue his postdoctoral work at Biozentrum University of Basel, Switzerland, at the University of Rochester, USA. He then joined IISc in 1989 as an assistant professor, then furthered as an associate professor in 1995, and then he was appointed as a professor in the year 2000. He served as a professor and chairman of the department. In 2015, he has been appointed as the president of Jawaharlal Nehru Center for Advanced Scientific Research, JNCASR, in 2015. Currently, he is serving as an honorary professor in the Department of Microbiology and Cell Biology at IISc Bengaluru. Professor Nagaraja has been a recipient of several awards, such as the prestigious Shanti Swarup Bhatnagar Award in 1999, J.C. Bose Fellowship in 2008, TWAS Prize in Microbiology. He was also awarded M. Vishweshwaraya State Award by the Government of Karnataka for his novel research contributions. Recently, he was also awarded the prestigious Goel Prize instituted by Kurukshetra University. Professor Nagaraja had numerous publications in journals of international repute and guided numerous PhDs. Apart from his scientific contributions, Professor Nagaraja is an ardent devotee of Bhagavan Baba, and he has been associated with the Institute and the Department of Biosciences in various capacities. He mentored faculty from the department and continues to mentor them, and aided in several students for carrying out their summer fellowships, internships, and research assistantships at his laboratory. Today, he'll be addressing all of us on the topic, challenges in tackling the resurgent drug-resistant tuberculosis. Professor V. Nagaraja.
You have to switch off these lights. Switch off right away. Yeah? Switch off this. Switch off the first row lights. If first row. Om Shri Sai Ram. Can you hear me? Hear me well? Too loud? Maybe this is better? This is better? This is even better? Okay, this is fine, right? So, good morning all of you. At the outset, I must thank Sai Ram first, and then the Department of Biosciences. So, Chair Professor Venkatesh and all the other respected faculty from different bioscience and other departments, including respected teachers from Anantapur campus and dear students. I'm indeed privileged to give this first endowment lecture or memorial lecture in honor, in the memory of Dr. K. Gopinath Rao. From whatever introduction Arun Sai gave, I gather that he is a very distinguished doctor, worked on urology and some of the other areas. Yeah. So the talk I am going to talk today has some connection with the medical field, also urology in one sense, but more generally it is a very, very important topic. So I hope it is befitting in the memory of Gopinath Rao, and hope, is it audible enough? Okay, if there is any problem, let me know. I hope it is a befitting talk on the occasion. So, both myself and my, why? You think I'm going to follow? Yeah, that's okay. I'll take care. Yeah. Until I might talk, I will not go. So, I'm indeed privileged to give this talk. As I said, so the title of my talk is Challenges in Tackling the Resurgent Drug-Resistant Tuberculosis. I will not make it too technical. Towards the end, I will highlight very, very little bit of our work, what we are trying to do. When I talk about challenges, it shouldn't be that you just talk about challenges, because it happens in many areas that people talk about challenges and they don't try to find the solution. So our minuscule efforts in that direction I will tell towards the end, not in elaborate fashion. But both myself and my good friend D.N. Rao, we are here today and tomorrow. Anybody who has any questions, please feel free to discuss with us. I should be able to answer most questions, almost all. If I can't handle, he is there. If he can't handle, I am there. And if both of us can't handle, you have the Wikipedia and other things to find out. But that may not be needed. Can you see the slides? Well, behind? You sure? Good. Okay. If you want, you can turn off these lights also. But right. So I'm not going to. On the left side are my PhD students, about 40 to 45 PhD students so far, but many of them have worked on this area. Not all of them, only those relevant I have put here. And these are my major collaborators for some of the work in the drug discovery efforts for tuberculosis. And we have been funded at one time by European Union Consortium, more medicines for TB. Anyway, yeah, that's much better. Right. So I will go on. Where do I go on? Yeah. Right. So tuberculosis is a very ancient disease. It's a disease, even Egyptian mummies, there in their brain or in the body there were lesions. It was identified. It's one of the oldest pathogen known. More recently we have the hospital acquired infections, other infections of the bacteria which are not even considered as pathogens. Whereas This is one of the oldest bacterium known. Possibly the human tuberculosis evolved from bovine tuberculosis, because there is a Mycobacterium bovis which causes cow TB, which is very, very similar. When I say similar, in the current day genomics era, in the DNA sequencing era, it is more than 99% similar, more than 99%. So Mycobacterium bovis to Mycobacterium tuberculosis, they are very, very close. Then the monkeys got infected, and possibly if we believe in that line of evolution, that we have one of the oldest pathogen which has learned to live with the host. It likes to live with the host. So almost all of us have this tuberculosis bacilli inside, but we don't come down with the disease. Those immunocompromised, those whose body defense is weak, those who have malnutrition or variety of other conditions, they end up having the disease. Most of us are exposed to the disease, one way or other, public transport, but our immune system is strong enough, it hides in our body. Discovered by Robert Koch or Koch, whatever you want to call, it is not gram-negative. Microbiologists here will appreciate what is gram-negative, gram-positive staining. It is detected by acid-fast staining. I will not tell the details of it at this moment. It is a different kind of stain to pick up the bacteria if you are able to see the bacteria. But the one which is hiding in the body, you can't see them. There is no way to detect them even now with all the discoveries, all the developments what we have. The problem now is, of course, it's not just a tuberculosis, and the treatment of tuberculosis is very, very daunting task. For a regular TB, tuberculosis which was infected, almost every family had some patients or other. My mother, when she was 13-year-old, she had, before her marriage, she had tuberculosis. She was treated by a doctor who married her afterwards. Okay, the village doctor. Okay, that's a story, long time ago. This is a so tuberculosis is a curable disease at that time. But now we have multi-drug resistant TB, MDR TB. You have extensively drug resistant TB, XDR TB. Then you have, don't worry Pradeep, then you have TDR, totally drug resistant TB. What is multi-drug resistant TB? It is resistant to the frontline anti-TB drugs. Whereas XDR TB is an additional, extensively drug resistant, additional drugs. Definition, exact definition I can give you later if anyone is interested. Then total drug resistant, they reported it few years ago in Mumbai hospital, means there is no drug which can treat total drug resistant TB. Okay? How do I have to learn this? Which way it goes? Yeah. This is next slide. Yeah, that's what I see. So now, many of you know TB bacteria infects lungs. It makes large lesions called granulomas inside the lungs. And then patient starts coughing. It's a different kind of cough, and the doctors can certainly recognize very easily. That is the most common TB. But what you may realize, ladies, gentlemen, boys, girls, is it infects every organ in the body. That's why even the urologist like Gopinath Rao would have come up with the TB of different, of that kind. So it infects, it's only partial list here. It can be brain TB, pulmonary TB is a common one, bone TB, gastrointestinal TB, eye TB, lymph node TB, miliary TB, and urinary tract genitourinary TB. And there is extrapulmonary TB, the skin TB, and many, many other things are there. It's not even listed here. Let me see whether it works now. Yeah, I hope it is, yeah. So, MDR and XDR TBs, they are all over. I will go through, I will slowly, I will go a bit faster as I go along. So, if I am not making myself clear, you can raise the hand. So, the color code shows the number of, what shall I say, the area where there is a higher TB incident. So, for example, India, Africa, this pointer has no life. The pointer has no life. You may have to move around there. Okay. So, the color code shows the different intensity number of is given there. But the point is 1/3 of world population is infected with TB, 9 million new cases, 20 million chronic cases of active TB, globally about 2 million associated deaths. So, disseminated TB makes up 10%, and the childhood TB is very difficult to detect. Even now with all the diagnostics, everything is known, it's extremely difficult to diagnose. So, there is lot of efforts to find new diagnostic tests, hundreds of them, and I am collaborating with somebody in Denmark now to make a new diagnostic efforts. I don't know how far we will go there. Many of them are not successful. So, you have the, I will come to the drug part now. So, WHO had recommended a treatment option. When you go to the hospitals here, you end up getting, for any infection, let us say urinary tract infection or some other infection, not the current resistant one, they give one particular antibiotic, one particular drug. You are supposed to take for 5 days or 7 days depending on the doctor, but after 3 days we end up stopping it. And that can lead to additional complications, drug resistance and other things. But for TB, drug sensitive normal TB, it's a 6 months treatment with the 4 drugs. 4 drugs. I mean, initially 4 drugs for 2 months, followed by 2 of them continue for another 4 months. So, it's a multi-drug therapy. When it comes to multi-drug resistant TB, it's even bigger problem. Yeah. So, this 6-month therapy is called short course chemotherapy. A short course chemotherapy involves 4 drugs: rifampicin, INH, pyrazinamide, and ethambutol. That is a short form, RCC form, right? Continuation phase, only rifampicin and isoniazid for 4 months. This is called direct observed therapy. This is a WHO recommended. 95% cure rate for drug sensitive TB, but for drug resistant TB, these drugs don't work because pathogen is resistant. It's extremely clever bacteria. You need 18 to 24 months with the fluoroquinolones or aminoglycosides, and there are new generation ones. And in fact, new drugs have come to the market, and there's a new combinations have come up. I can tell you about it later on if you wish to. And there is a 18 to 24 months, only 50% efficacy. Right. So just X-ray was the, there are spots there. Doctors can easily find out the lesions. That is the most authentic test. But now there are newer assays, Gene Expert and other assays for the detection. But none of them are 100%. At best, they go up to 90%. So 100% is not there. I know of my colleague in Indian Institute of Science. He had an ear infection. They treated with and a fever. They treated with every other antibiotic. Nothing was working. Finally, they treated with the combination of 2 anti-TB drugs. It got cured. So it is a not diagnosable TB suspicion because it was not cultured. Then in bone marrow transplantation, the senior citizens like me, when they get a bone marrow transplanted or something, they come down with very high fever. So some young doctor from St. John's wanted to come and interact with me and pick up, suspected is tuberculosis, because only way that fever was treated was by anti-tubercular drugs. That was a reactivation of a hidden TB. That's a suspicion. Nobody has proved it. So I said, okay, let us start the work. But then soon he got a fellowship in US, he went off. So collaboration ended right there. Okay, that's a sad story of it. So very clever pathogen and tough pathogen to work with. Escherichia coli, some of you work here, microbiologists, once in 20 minutes it divides in the lab, whereas this is once in 24 hours in the lab culture. Okay, extremely slow growing, and multiple fortresses, lipid and other complex lipid polysaccharides, and when it infects the host inside our lungs, it multiplies, takes the resources from the neighboring cells, and neighboring cells, there are biochemistry students here, chemistry students here, all inside our body, every pathway is cellular pathway is a biochemical pathway. So you have energy generation inside the body. The primary is a TCA cycle, glycols TCA cycle. That's what TB bacteria does outside when you are growing in the culture. But intracellularly, it changes into a different kind of life cycle, involving lipid oxidation. So it borrows the lipids from inside, totally different lifestyle inside. So it has got different lifestyle. This is something you need to know. And because of that, it can borrow nutrients from neighboring cells, and neighboring cells get lysed, and as a result you end up with TB cells multiplying, and that forms something called granuloma, and basically our host defense cells, everything gets manipulated by this. And I will not go into this much. Basically what happens is when a pollen or a virus or bacteria is taken in, they are engulfed by our macrophages, defense cells, and inside the macrophages they get fused into a vacuole, and the vacuole releases all kinds of enzymes, and to degrade it, the virus or the bacteria's carbohydrates, lipids, nucleic acids, different kinds of enzymes, hydrolases, proteases, lipases, others, and that way then it can make from those antigens the immune response can be generated. But that kind of a fusion is prevented in case of TB. TB is a clever bacteria. It overcomes this fusion with the lysosomes, bacterial inside the cell. As a result, it is able to survive inside the host. As I said, it is a very ancient bacterium. It's a very ancient bacterium, one of the cleverest bacteria. I can tell entire Mahabharata about TB, but I will not say, just take my word, found many ways to survive. In fact, more recently was shown a new way it can survive, an extremely new way, published in a very good journal, and can stay dormant inside the healthy individual like us, difficult to handle. Emergence of extensively drug resistant, multi-drug resistant bacteria has created an even bigger problem. And our Venkatesh sir talked about the pandemic. During pandemic, see, our healthcare system is very good for TB. In South India, in many states, southern states, there is a very active DOTS program, and that's how it was controlled. But during pandemic, not in all states in India it's such good, but in some states it's extremely good. And TB healthcare providers, and as long as you go to government hospitals. But then during the COVID pandemic, all of them are channeled for COVID. As a result, the tuberculosis patients were neglected, and its incidence has gone up. But by and large, there is a very good controlling system. As long as you don't go to some private quakes you go to the regular hospitals and you take the medicine properly. Difficult to handle, and there is a terrific collaboration between HIV and TB. Lot of HIV patients, AIDS patients die of tuberculosis because their immune system is down, and as a result tuberculosis takes over and they die of tuberculosis. It's a global challenge, so you have to find a way of counter the resurgence, and that has been going on for ages, but I think we have not been successful. So as I said, most formidable pathogen, no available drugs can kill latent TB. The hidden TB, no drugs can kill. This is the problem. So when the 6 months therapy, you almost eradicate it, then your immune system takes over, so it's almost eradicated. But the reactivation takes place in healthy individuals, as I said. It can, but it's almost 99.9% cure, and it will unlikely to come back as long as you remain healthy, right? And then it becomes dormant. There is no way, no drugs can penetrate. Drug resistance is due to changes in the physiological state in bacteria, and many other challenges. I will not go into because we are running behind schedule at this moment. So 6 components to stop TB. You have to pursue this in every state. In Indian context, you have to pursue it. Now that COVID is under control, in many TB healthcare workers have come back to work to do TB work, but in many states it's not the same way. And address TB, HIV, and MDR TB, and in the vulnerable populations. You need to deal with them. Contribute to health system strengthening in the primary health care. Engage all healthcare providers. Empower people with the TB and communities through partnership. There is NGOs and many things are happening. But enable and promote research. That's where people like us come. We end up working on TB, doing research, trying to understand the pathogen, and so this is also important to find drugs or vaccines. When you talk about vaccines, BCG was the vaccine when all of us or almost all of us were not effective. Actually, in our population, it's not very effective, but okay. But in Caucasians, it's a little bit more effective, but still not very good. And none of these vaccines are of a high efficacy. But new generation is being researched, and many other vaccines strategies have come up. Some of them are supposed to be more successful. So, for example, and TB is not just one vaccine the way you give for some of the things. Some of the other viral diseases, for example, you have one vaccine, that's it. Here there are different ways: vaccine to prevent infection, conventional protective vaccine, or booster vaccine, or you can have a therapeutic vaccine, then you can have facilitated chemotherapy by vaccination, or vaccines to prevent reactivation. So all these strategies are being done, and now, yet vaccine may not be an effective strategy in my opinion, simply because the TB bacteria is not only clever, it comes in different forms. I will explain in one or two slides in the next slide, I think. Hopefully it's there. Yes. TB vaccine is not very good because there is a tremendous heterogeneity. It is not like a Mycobacterium tuberculosis, it's a one species, fine. From Mycobacter genus, Mycobacterium tuberculosis. But if you look at the strain isolated in Beijing, strain isolated in Africa, strain isolated in South India, each one has a different degree of virulence. And there are thousands of TB strains have been sequenced now, genomic sequence. Genome sequences, DNA sequencing has become very, very easy these days. I am showing a slide. There is a tremendous variation. Then they have single nucleotide changes called SNPs, or they have large chunks deleted. For example, laboratory strain H37RV. We all work in the laboratory. We means my students go to the BSL3, Biosafety Level 3 facility, work there wearing masks, gloves and special dress, because that's how we can work with TB these days, and many of them do that. So BSL3, that is a H37 Mycobacterium tuberculosis H37 RV. But then there's the RA strain, A virulent strain. These were there when I was student days. These are the old laboratory strains. A virulent strain has a large deletion in a locus called RD10. So, what I'm trying to show you is large variation. I will not explain this bioinformatic thing. Basically to say each bacteria comes differently. So the vaccine may not be efficacious when there's a large heterogeneity in the bacteria. What is effective here, what may not be effective. That's why BCG is being more effective in Caucasians, I said. The reason for this is MTB, many other bacteria also, they can duplicate, they can delete by many mechanisms as shown here, right. So, drug is a way forward. In the last 10-15 years, there is a renewed interest. There was no interest on TB in Western world, developed countries, because they thought it is a poor man's disease, all India, Brazil and all those places. But now things have changed. In Sweden, people are dying of tuberculosis, migrants from maybe from Eastern Europe. In healthcare workers in New York have died of tuberculosis. Because people who came to Ladakh or somewhere else trekking, they got reactivated when they are old and they got infected, healthcare workers. So TB is a global problem now. And multiple drug resistant to TB is a global problem. So new generation drugs have come up by big consortiums. So I was also part of one of the consortium. I will quickly try to finish because I am only spoken for so far 25 to 30 minutes. We started late, but in 5 to 10 minutes I'm going to wind up. So we work on TB bacteria to find new drugs. Many of my students have worked on this. What we do? We do biochemistry. We work on the enzymes in the lab. TB enzymes, study their properties and try to find their weakness. Try to find the weakness. And so when you study all its properties, you know what property you can tackle. Develop small molecule inhibitors. So we work on enzymes called DNA topoisomerases. We work on, these are topoisomerases. We work on RNA polymerase, the enzyme which makes DNA to RNA. We work on transcription factor, termination factor. One stops transcription. We work on another transcription elongation factors like GRE and others. So many, and then also we work on proteins which are not enzymes but which decorate DNA. They hold the bacterial DNA, bacterial chromosome, bacterial nucleotide together. All this we work to understand the biology of the bacteria. To understand the biology of the bacteria, then you understand its weakness. This you do with every possible approach: genomics, transcriptomics, biochemistry, microbiology, immunology, structural biology. Name the branch of science we employ to do what we do. Right. So next few minutes I will tell you what is a bacterial DNA looks like. It's a textbook information. I don't have to tell you. This is a classical textbook picture. Highly compacted DNA is opened up when you lyse the cells. Okay. But why otherwise it is compact inside the bacterial cell? Because of the bacterial chromosome is called nucleoid because it doesn't have a boundary, it doesn't have a nucleus. But then within there you have enzymes which make them compact by supercoiling called DNA topoisomerases. I will not elaborate much here. And also you have decorating enzymes originally called as histone-like proteins. Now we call them NAPs, nucleotide associated proteins. They bind to DNA, bend the DNA, write the DNA, twist the DNA, do all these things to keep the DNA compact. These are called NAPs, and that's what they do. Now it has come to life now. So these are those proteins, different color code. these are naps, right? So topoisomerases, they are considered as the magicians among magicians in by the ultimate authority in this field, Jim Wang. They bind DNA, cut the DNA, pass the intact DNA across and reseal the DNA, and different classes of the enzymes. And as a result, inside because it's such a compact, you know, it's so easy to get telephone wire or some other wire get into a tangled mess. So DNA will get into the mess. Those messes are sorted out, not a DNA is completely sorted out by topoisomerases. Importantly, during the process of DNA synthesis and RNA synthesis, the double-stranded DNA has to be opened up. When you open, there will be more coiling ahead and undercoiling behind, positive supercoiling and negative supercoiling. That happens along with the transcription, along with the RNA synthesis. So DNA to RNA is called transcription, RNA to protein is called translation. Yeah, they all know. So this transcription and topoisomerase go hand in hand. RNA polymerase is doing RNA synthesis, then ahead is one of the topoisomerase removing positive supercoils, and negative supercoils are removed by another topoisomerase. And we work on all of them, all three of them. Because the topology and transcription are coupled, the terrific nexus, they go hand in hand. Now our new strategy is to find drugs for RNA polymerase. You have rifampicin already. Rifampicin is a drug for TB treatment. Along with that, now topoisomerase inhibitors, combination. Right now in my lab we are trying that. Our own new candidate discovered. So this just to tell you in a glimpse. So there are many, hopefully there are some chemistry students. Before you sleep, there is one chemistry slide. The what you want to show you is, biochemistry is all, you know, life is full of, life is because of a lot of biochemical reactions inside the body. Make it oversimplification. But now, the topoisomerases are magicians simply because the kind of chemistry they do. This is a very, very interesting chemistry. What they do, these enzymes, they bind the DNA. Afterwards, they cut the DNA like restriction enzymes. Restriction enzymes stop then and there. After cutting them, there is a tyrosine. Tyrosine is an amino acid, as all of you know. Tyrosine hydroxyl makes a nucleophilic attack and makes a covalent linkage with the DNA. If it's making a 2-strand cuts, covalent linkage, one here, four strand over like a restriction enzymes. And there's a covalent, protein DNA covalent link. Then through that opened DNA, intact DNA passes through strand passage. Afterwards, the free OH, after the strand passage, it reseals by second nucleophilic attack, second transesterification. 2 transesterification reactions in between DNA strand passage. That's how the overcoiling is removed by the action of topoisomerases. Only these enzymes can do it. You can go and see a movie, very, very beautiful movies are there for this. If I didn't make myself clear, I can explain again later on perhaps. But the point is, now you can find the drugs which arrest the reaction. If you arrest the reaction, DNA is cut, it cannot be resealed. If the bacterial DNA is cut, it's gone. So, topoisomerase, fluoroquinolones, and many other drugs, what you want is, they arrest this topoisomerase reaction. As a result, DNA is cut. After first phosphodiester cleavage, DNA is cut. It is not resealed. It is not religated. At that step, you target it. Our idea was to develop this kind of drug for the topoisomerase 1, and we succeeded in our efforts in the MM4 TB. And that's what I'm showing you. Topoisomerase 1 had no drug. We found these small molecules by molecular docking. Structure was not known. Now the enzyme structure is solved, and we find these small molecules. What are these drugs? These are called imipramine. They are antidepressants. In 1950s in Swiss Alps, in the TB sanatoriums, they were giving this for TB patients and they were recovering. But they were given as antidepressant. Nobody knew it can work on TB. Even now we don't know it works on TB. But it binds to the top enzyme what we work with, and binds, and we have shown it inhibits specifically mycobacterial enzyme, not other bacterial enzyme. So that's good for me, good for us. So as a result, it's a case of repurposing of the drugs. Many major companies are trying to repurpose the drugs now because they want new drugs for different diseases, right? And this is just a slide. I'm not going to show 10 seconds only. Basically, we show that M. tuberculosis gets killed. That's what's shown here. And now we also targeted similar way histone-like protein, and we solved the structure of one of the major one, and which binds to DNA. DNA binds here. So if we found a molecule which displaces DNA, then it can be inhibited. And that's what we have shown. These molecules are developed. It displaces the bound DNA by experiments. No, we end up doing a lot of experiments. Only I do the talking. My students do a lot of, that's why we end up publishing lots of papers. So what I show here is the nucleoid property. Nucleoid is compacted, but when you treat with these molecules, the compact DNA, the DNA is stained by DAPI. It gets DNA spread all over, and in electron microscope you see. As a result, the DNA has to be kept inside in the cell in the right place, not everywhere. As a result, cell killing, lot of cell killing. So, okay, and so this is also a possibility. This also we are trying to combination. Look, this will not become a drug, but we can try in combination with others, like the previous one against the topoisomerase inhibitors, right? So, but we also do, to know how this is inhibiting, do the transcriptomics. Look at what all RNAs gets affected. And by treating with this one drug, we know that this particular protein, small protein which decorates, affects all pathways in MTB. So I am towards the end of my talk. So TB desired drug profile, I come back to this. We have not solved the problem. Sensitive TB, 6 months therapy, I said. Anybody finds a treatment which is able to cure TB in 2 months, Nobel Prize. New combination drugs. One drug will not work, nahi chalega. It's a Nobel Prize. Even now, 6 months is therapy. Newer combinations come, they are trying to make it for 4 months. Three drug combination. It has come. I don't want to overload you with all those names. So you have ideally you have to have oral delivery. If 6 months you can't keep getting a pricks, or intravenous or whatever it is. And if it has to be working for extracellular, intracellular persisters, and there should be, when you work with 3-4 drugs, there should be no antagonism. There should be synergy. One drug should not work against the other. Then compatibility for antiretroviral therapy, because TB, okay? And toxicology acceptable. Therapy ideally results in less than 6 months, 4 months, 2 months. That's a dream. That's still a dream. People are in India, there are a large contingent of TB research. Elsewhere in the world, it's a big challenge. So there are many barriers, including financial, and the big companies have stopped doing research. So this is the last slide. Your the battle tank, MBT, NTB. Very similar, only one word different if you are looking at the alignment. Both are formidable. But MBT is more easily destroyed. TB is not easily destroyed even now, though there is therapy. This is the take-home message for you. Both have weapons for attack and destroy, very heavy armor, defense systems to protect itself from the attack. And we have found a new defense system more recently. TB protein goes into the host and changes the host chromatin, goes into the host nucleus, changes its host immune system, because host immune system wants to attack. So there are it has so many new strategies, so many virulence factors. I can go on speaking for one whole week about TB's this thing. So I must stop you here in the interest of time. Thank you for your attention. Sri Sai Ram. Any number of questions, but I think he has to talk about any number of questions. Yeah. I don't want to chat until I go down. Yeah. Take it away. Okay.
Thank you, sir.
Yeah.
Thank you so much for your valuable talk.
So he's asking any questions. That's what I'll shorten it. Anybody has questions, please raise hand. Yeah, and come to the mic or something and identify, please. Please hand over the mic for my young friend there.
Saram sir, thank you so much for your insightful lecture. My doubt is like a person affecting from a TB and a person who is not affecting from a TB. So when we come to the DNA sequencing of that person, so what is the difference between the DNA of that person affecting of TB and healthy person?
You will not find much difference at DNA level. I can elaborate during lunchtime if you ask me, if there are other questions. What I said is the mycobacterium bacteria genome sequencing has been done. Human DNA sequence between suspect one who got infected versus not will not be such a big difference. It is just that the immune compromised state, that is a more propensity. It's not only immunocompromised, even persons who are healthy can come up if there is a large bacterial load, come down with the disease, if there is a large bacterial dose. Okay?
Yeah. And sir, like, is there any some device or something which can detect TB at its early stage?
That's the point. Yes. That's a very good question. That's a challenge even now, as I said, detection early. And childhood TB is extremely difficult to detect, even now. And childhood TB is rampant in India now. And that's not pulmonary TB, very difficult to detect. That's why a lot of new diagnostic things have come up with much better accuracy. Still, it's a challenge. So it goes in empirical fashion. You know, when nothing else works, you start treating with the TB drugs, when you have high fever and whatnot. As I said just now, you know. So, but many newer, if you look it up, you will find many new diagnostic ones. People have tried to do diagnostic from urine. Now people are trying to do it from saliva. And, you know, but, you know, you should have enough sample sensitivity should increase. Okay, and you know, you'll have a lot of false positives, and it's a challenge.
Thank you, sir.
Yeah, but you know, for a manifest TB is very easy to detect. You know, but the way patient coughs itself, and then you know you can culture it. It takes long time to culture, but you know, chest X-ray, and that's but even they can be cured if it is drug sensitive TB.
Yeah. Thank you, sir.
Huh? Over. So Pradeep says no more questions, but you can ask me during lunchtime.
So much, sir, for your informative talk. I request the house to join me in another round of applause for Professor V. Nagaraja. Now, I request the director of the campus, Dr. Raghunath Sharma sir, to hand over a token of love and appreciation for Professor V. Nagaraja. Thank you so much, sir. Now we have Anusha ma'am, Assistant Professor, Department of Biosciences, SSSIHL of Higher Learning, Anantapur campus, to introduce Dr. D.N. Rao.
I have the pleasant duty of introducing Professor Desi Razu Narasimha Rao, Honorary Professor at the Indian Institute of Science, Bengaluru, as our guest of honor for the International Microorganisms Day. Professor D.N. Rao obtained his PhD degree from IISc Bengaluru in the year 1982. He then embarked on an illustrious postdoctoral journey spanning across various institutes like National Institute of Health in the United States, Biozentrum in the Switzerland, and University of Cambridge in the UK. He then returned to his alma mater, IISc, in the year 1989 as a faculty in the Department of Biochemistry. Later, he was appointed as the convener of Talent Development Center at IISc Chellakere campus. Professor D.N. Rao is a distinguished biochemist known for his groundbreaking research on DNA-protein interactions, particularly in Helicobacter pylori and Neisseria gonorrhoeae. His work has significantly advanced our understanding of DNA methylation and gene regulation. He has authored several papers which were published in journals of international repute. Beyond his scientific achievements, Sir is an ardent devotee of Bhagwan Baba. And his long-standing association with our institute has enriched our department, inspiring both faculty and students. We are privileged to have him with us today. Please join me in welcoming Professor D.N. Rao to deliver his talk on the topic, Exploring the Invisible Universe in Us, the Human Microbiome. Professor D.N. Rao.
Ah. Yeah, yeah. Yeah, yeah. I can move around. Yes, I'll finish. Om Shri Sai Ram. Thank you very much for inviting me to give a talk here. I have first come here in 2002, 22 years back. It's always a divine experience to come to Puttaparthi. And nothing has changed since then, except that the students are different, the enthusiasm is more, and a lot of teachers from the other campuses are here. So it's a pleasure to be here. And I thought I will speak on a very general topic, something called the microbiome, the human microbiome that I plan to talk about. So we can start that and now First of all, I would like to impress people on this topic, that the microbiome is like an organ, like the liver, like the kidney, like the spleen. The microbiome, the human microbiome, is also an organ, a forgotten organ. And it's one of the most important organs. All organs are important, but this is one of the most important ones, and a lot of work has been done in the last 15, 20 years on this particular organ. And that is what I call this titled as Exploring the Invisible Universe That Is in Us. When we talk about, or anyone talks about universe, we imagine the hundreds and hundreds of stars that we see in the night sky, typically. And nothing is more astronomically fascinating than the invisible, the universe that we know of. But within us, within the human body, within you and me, there is a universe that exists. And I tell you, this is a universe because of the numbers that we are talking about. 100 trillion microbes are present in each one of us. 1 followed by 14 zeros is a number of organisms that you have, microorganisms that each one of us have. Roughly a 70 kilo weighing person, if you take all the bacteria, all the microorganisms within you, it weighs about 1.5 to 2 kilograms. The liver has a weight, the kidney has a weight, the microbiome also has some weight. So it is the existence of all these microbes within you is what makes this whole area of microbiology very, very fascinating. And that is what we want to impress upon, and I want to talk to you about these things. All right. My slides are all blue shade, unfortunately. The number of bacteria, the number of organisms that are present in each one of us, that number is anywhere between 10 to 100 times more than your own human cells. The number of human cells that we have is much less compared to the organisms that we have. And therefore, you have to question yourself, are you a microbe or are you a human being? And therefore, I picked this up very nicely from an article which says, I have to allow myself to introduce me, myself and us, the bacteria. We all constitute what you call as yourself or myself and things like that. And this is what I want to talk about, the microbiome, the existence of this within us. So we forget these slides, they're not properly done here. A microbiome is a living organism. A composition of living organisms within you. If it's within you, it's a human microbiome. If it's within a snake, it's a snake microbiome. If it's a cheetah microbiome. An elephant has a microbiome. A plant has a microbiome. It's all the ecosystem that is there within the living organisms. And it usually consists of mostly microorganisms, some algal species, some fungal species, some prokaryotic invertebrate members, and these all constitute what we call as the microbiome. And the microbiome is very, very fascinating in the sense that together all these organisms in the human body, a community of microbes is called a microbiome. And it becomes now very important that these organisms, the 100 trillion bacteria or microorganisms that are present in you, are important. Now, experiments with mouse is not possible with human beings. That people have done where they have taken most of the bacteria out of a mouse, for example, and then said that these are the effects if you don't have the microbes within you. You can't do such an experiment with a human being, and therefore most experiments have been done with mouse and rodent models. And from that kind of studies, it's now clearly established that the microbes, that the 100 trillion bacteria that are present in you are important. We'll come to that later on when I give you some details of that. So microbes are all over us. There are, as I said, 100 trillion of them, and many of them in all parts of our body. External, internal, everywhere you have microbes. You have maximum on the skin, you have inside in the intestine, in the pancreas, you have in the gut, lungs, skin, eye, all kinds of fluids that the body has have contained a lot of microbes. And therefore, the existence of a normal human being, so-called normal being, is you have to appreciate how these bacteria all live together and then survive in this particular environment. So here you have just a picture of this to show you that you have bacteria in every part of your body, external and internal. Tomorrow you can do an experiment, you can go to the laboratory, take an agar plate, a LB plate, just swipe from the inoculating nebular from your skin and plate it, and the next morning you will find plenty of bacteria, microorganisms that come. From any part of your body you take and do it, and you have tons and tons of bacteria, tons and tons of microorganisms. So it just shows you that this is a very important organ, and this helps in the homeostasis of a human beings, and that is what I'm going to talk about. All right. Now, two things that become important when you study microbiome, the abiotic conditions, and that is the pH, the temperature, the oxygen level, and the moisture. All these affect the kind of microorganisms that are present. Whereas if you take the biotic factors, then you have the types of cells, the secretions of the humans, and the presence of the other microbes that are present here. And if you study, and most of you study microbiology, the classification, you have all sorts of bacteria and you have all sorts of microorganisms. Some are bacteria, some are archaebacteria, some are eukaryote. The fungus, algae, all present in your body. So it's a variety. There's a whole extreme representation of the, you can see evolution actually inside your human body. Now, because of DNA sequencing and the kind of technology that developed, you can actually isolate microorganisms. Not microorganisms, but can you extract DNA from any particular tissue. For example, if you now take the mouth, for example, you can isolate the DNA and analyze by DNA sequencing, and because technology has become so advanced and easy, you can actually identify organisms based on DNA sequence. So especially when you talk about bacteria, these are the different kinds of bacteria that are present in the human body. And that is, you have actinobacteria, you have the firmicutes, the bacteroids, the proteobacteria, the fusobacteria, and all that. There are different colors. And if you now take the mouth, if you take a swab from the mouth and do DNA sequencing, you can actually identify all these, the proportion of the bacteria that are present in the mouth. You see, for example, the red bacteria here, the firmicutes are in large amounts, whereas the green color is actually the proteobacteria, and you have some of them. So every organ can be analyzed to find out what kind of bacteria. And that is what is the microbiome analysis, which is now becoming a very routine feature in hospitals and in diagnostic field. Find out what kind of organisms that you have in different parts of your body. And this is a nice representation to show you that if it's mouth, then here in the esophagus, if you look at, in the esophagus, you see there's so much of red here. There is so much of firmicutes here. Whereas you come to the small intestine, half of it is green, which is proteobacteria. So every organ of your body has a specialized place for special bacteria. The large intestine, between the two intestines, the small intestine and the large intestine, see the proportion of bacteria, the kind of bacteria that are present are different. These are inherent bacteria, and they are important for that particular organ. You take the skin, for example, lots of actinobacteria, the blue color, for example. And similarly, if you look at the other organs, or if you look at the nasopharynx or oropharynx, or even the genital organs, for example, the composition is quite different. And therefore, this kind of analysis becomes important, because if you know the kind of bacteria you have, or you had when you're normal, or when you're sick, for example, if you now do the analysis that I'm sick and therefore my lung has now different kind of proportion of bacteria, and then you will know what kind of antibiotic you should use. You must use a drug or an antibiotic which is specific for actinobacteria or the firmicutes or the bacteroids and things like that. So personalized medicine now becomes very important, and it's very easy to do because you have done the analysis. So you know when you're sick, this is the proportion of the bacteria which has increased compared to when you were normal. And that is where the microbiome analysis has now taken off in a big way. And many Western hospitals, maybe starting in India also, that the microbiome analysis is done just to find out. And you can't just take any antibiotic and take off erythromycin and swallow and think everything will work. If you know the kind of bacteria, you can then target that bacteria because of the nature of the antibiotic and things like that. So that is where the microbiome analysis comes. And the microbiome analysis is nothing but to know what kind of bacteria you have. When you're a child and when you're adult, your bacterial composition can change. And it does change. And we'll talk about that later on. If you look at families, for example, my family, for example, if you analyze my grandfather, father and things, there are microbes which are very characteristic to our family, the proportion of it. So it is this kind of analysis that has helped many doctors to analyze and cure people of bacterial infections. So that is what it is. So how do we acquire this flora? From where do they come? Or everything comes from the mother, right? Because the baby is born there and therefore baby grows up there, and the dispersal from the delivery and the mode of delivery of the baby becomes important. If it's through the vaginal, if it's a cesarean, the composition is different. So therefore nowadays doctors usually come back and ask you, how were you born? Was it a C-section or was it a natural? Because then they would know what kind of bacteria you have and therefore treat accordingly if you have infections and things like that. It becomes very, very easy now for doctors to analyze and give you a proper treatment if they know what is the composition of your bacteria. And that emphasizes the point that the bacterial composition is very important, is very critical, and now it has become such a hype that if you are smiling, the composition is different. If you're crying, your composition is different. Everything is attributed to the composition of your microbiome. So a lot of studies have been done. You have to take them with a pinch of salt. People have done things like, which is very valid, which is true, that children from rural areas or children from cities who use McDonald's, French fries, pizzas, their gut composition is quite different from the people who eat ragi and things like that. So there's a lot, diet does influence your bacterial composition. And therefore these kind of comparisons have come up and the whole area has actually boomed just to tell you how important this is taking place. From feeding, breastfeeded child versus children who are not breastfeeded but fed by milk, by bottles, their whole composition is quite different. And therefore, these kind of studies have revealed useful information as far as that is concerned. I'll skip them out this second. What are these 100 trillion bacteria doing in you? Why are they there? For a variety of reasons that experiments can't be done with human beings, it has been done with mice, with mouse models. And from such studies, people have found out that there are two vitamins here, vitamin K and vitamin B12, which is very important for us, for clotting purposes, for anemia and all sorts of things. These are not made by the human cells at all. They're all made by the bacteria that are present inside you. You can't get it from plants, you can't get it from yourself, but the bacteria inside you produce. So that is the main purpose of these bacteria. The other one, which I'll highlight, is they stimulate the development of certain tissues. When your intestines are developing or certain lymphatic tissues are developing, the bacteria help. So in other words, in the mouse, what they found that they moved the bacteria, intestinal development didn't work, lymphatic system didn't work, and that is how we know the importance of these bacteria, for example. I will not go through the rest of it, but these are important. And this is where the paper came out in 2001 in Science, when they had a mouse which was raised in a germ-free environment. No germs at all, no bacteria. And that particular mouse had decreased nutrient absorption, less developed intestines, vitamin deficiency, underdeveloped immune system, very important, and they were highly sensitive to pathogens. So the importance of microbiome came out from these kind of studies. Look at your digestive system from the mouth all the way to the end. The pH in the whole alimentary canal is varying like this, 1.5, 5, 7, 7.9, 5.7. And if people actually estimated the number of microbiome, the cells that are present here, and this is what it is. The microbial biomass is varying here. You look at the large intestine and see the huge number of cells. So many microbacteria are present in the colon and in the intestine. So the pH dependence and in the number of cells are related to a large extent. In the stomach, for example, the stomach pH is very acidic. It has pH 1 or pH 2. Even in this condition, there are bacteria which survive. Helicobacter pylori is one such organism which survives under very acidic conditions. It is not an acidophile. It doesn't require acid to grow, but it survives there. It has a very nice strategy of surviving in highly acidic conditions. And the strategy that this organism uses, this is just one organism, it produces an enzyme called urease, which hydrolyzes urea, which is NH2CONH2, to carbon dioxide ammonia. That ammonia that is generated neutralizes the HCl that is created in the stomach, and therefore it survives using this very nice strategy. So if one bacteria like this has one strategy like this, imagine 100 trillion bacteria in you, the strategies that these bacteria employ. And that's the whole fascination with microbiology and microbiomes. Exactly what Professor Nagaraja mentioned about Mycobacterium tuberculosis, it's a real challenge. It's a real challenge to study and to find out how these 100 trillion bacteria are actually surviving, how do they affect their existence with the human cells. Remember, the human cells are, because we are all made of human cells and things like that. So I'll skip some of this because these are, I just mentioned about. All right. One of the nicest things, I mean, a very popular thing that came out that from these kind of studies in the early 2000, that there was some relationship, again done in mouse experiments, a relationship between obesity and the microbial composition. You have genetically obese mice and lean mice. And if you analyze the microbiome in both these, they are different. The amount of firmicutes is more or the proteobacteria is more, but there is a distinct difference between a fat mouse and a lean mouse. What is more interesting is, scientists have actually taken the bacteria from the fat mouse and fed it to the lean mouse, or vice versa, lean mouse or fat mouse. And a fat mouse became thin, a lean mouse became fat. Very interesting that the microbiome actually dictates obesity. So lots of people, including me, wanted to try out if you could change the bacteria so that I'm not so fat and it become thin. But these kind of studies have led to the importance of the microbiome. So very nice experiments that these people have done. You see the firmicutes and the bacteroids are different in obese mouse and in lean mouse, and you can see the mouse here. I will not go into too many details because we have lack of time. So the gut becomes very important. The diet that you take also dictates. And as I said, people have studied vegetarian diet versus people who eat meat. The composition is different. Not one is preferred over the others, but the composition varies. A fiber diet introduces different kind of bacteria, a non-fibrous diet. So it's a combination of diet and your own genetics that matters, that dictates this. Now I'm going to tell you a story about this couple here called, in America, a husband and wife staying in the same house, having only one kitchen and eating the same kind of food. But the lady had a big problem of diarrhea. And diarrhea is very painful. It's not just going to the bathroom and coming, but you have a lot of pain because of diarrhea. And she tried a lot of medicines, but it never worked at all. Just about that time, the microbiome research came out, and the local doctor there in the US, in Tennessee or something, said, let's change your diet. She told the wife, the lady, that since both of you are eating the same food in the same house, it's not the diet that is important, but let's transplant the microbiome from the husband and put it into the wife. And within a week, she was completely cured of her diarrhea. The diarrhea she had was because of an organism called Clostridium difficile, which is a deadly microorganisms. So your composition, if you change, then you can get rid of whatever illness or things like that you have. Now, the problem with these kind of experiments were done in mouse. It is very difficult to imagine how to take the microbiome from one person and give it to the other person. It's not easy. And the only way that you can do is through feces. The feces excreta has a lot of bacteria. So you must take the feces of the husband and then give it to the wife. And this is how it's called as the fecal transplant experiments. A lot of studies have been done now to show that the fecal transplant experiment works very well. If you guys are all very young, but earlier we used to have a Prime Minister, Morarji Desai, who drank his own urine for the simple reason of this kind of microbiome composition being altered and things like that. So it's come back in a big way, but we know the science behind it and things like that. All right. We come to another discussion on microbiome is something called probiotics. You must have heard in lots of, all of us eat yogurt, curds, perugu. Has a lot of nice organisms there. Of course, the lactobacillus casei, things are important. But a combination of these bacteria are now fused into a tablet, and if you take that, it seems to help. Probiotics. So a lot of probiotics now exist in the market, and these are the typical organisms that are present here. I've listed some of them here, but this is what it is. So you have lactobacillus, you have the acidophilus, the brevis, the delbrueckii, all these things are now put in a tablet, and you take that and it seems to help. But it's always good to consult a doctor before do some of these things. We also have, probiotics which are good for certain kind of diseases and things like that, but we will not talk about that. We also have something called prebiotics, that you take the organism, it is metabolized, and that metabolite then works on as a probiotic. So lots of research just because of the human microbiome, the nature of the human microbiome, that people have figured this out. So lots of these kind of drugs are now available. They are harmless, they are bacteria, but you must find what is the composition in your system and then take some of these things that you have. So talking about the microbiome, the twins, identical twins for example, the DNA sequence is same and all, but the microbiome can be different. So intelligent children versus non-intelligent children, all sorts of studies have been done. You have to take all these results with a pinch of salt. But some of these things, a lean diet and a fat diet, that seem to correlate very well with this. People with, children from Europe and rural Africa, where people have actually studied, they found a difference between the microbial compositions. These two slides are from actually Nagaraja's talk, which he gave some years ago that I bought. So the human milk has a lot of, so it's, breastfeeding becomes very, very important because the kind of bacteria that the child gets in the first stages is very important here. So this is what I want to conclude now, saying that if you just imagine the skin, take a section and people have done. This is in the early life, this is in the steady state, this is the barrier breach, and then in the late. The skin composition also has changed over the years. The bacterial composition has changed over the years. So these kind of studies tell that old people, aged people, their microbiome composition is different, and that is why their physiology becomes different. A lot of such correlations are there. One other correlation which I thought I'll mention is this, which is coming up in a big way. There's a depression that humans have and the stomach. Remember, the stomach and the brain are connected in more than one way. If you eat, if you starve. If you eat well, then you're in a good mood. If you starve, you're not in a good mood. This mood business that you talk about is all related to this brain axis, brain-gut axis. And people have found that the composition of this varies and seems to affect this particular thing. So microbiome is becoming a big study, is already a big study, and lots of studies to connect these kind of physiological processes that you have, I was talking about. So what are these microbes doing? What is the identity of the microbes? What are the forces that maintain the equilibrium? Because they have to exist with the human cells. Remember, they are more than us. And how is the host responding to them? And what is the characteristic feature? This is where all the questions in the microbiome research are being addressed. Lots of useful information is coming. It is not, I find that Pradeep does a lot of sequencing, so this is what is done. So some of these things can be easily done here. So I'll end the talk saying that, if you have to give an acknowledgement at the end of the talk, you can write like this. I would like to thank my director, my friends, and my family, and of course the huge mass of gut bacteria that I have here. Without them, I wouldn't have been here. So you have to worry about whether you're a microbe or a human being, and the distinction becomes very, very less. So thank you very much. I think all right, a couple of questions because we have to end now. Yeah.
Strictly a couple of questions.
Any question? You can ask me later on also. After you eat food, your mood is better. So you can ask.
We have time for 2 questions. If anybody wants to ask any questions to sir, maybe they can
Some
ask.
questions from Anantapur campus. It's always nice to hear from them.
When you transfer the microbes from a obese mouse to the lean mouse, the food intake, does it change?
Very good question. Yes, it does. It does. People have studied that. Yeah. Many of the physiological change, parameters have changed. Food intake is one of them. That's a good point. Right. All right. We can always discuss at lunchtime or till tomorrow and things like that. Thanks. I'll stand here. All right.
I would like to request the House in joining me in another round of applause for D.N. Rao sir. There is a lot of mystery that is still pending to be discovered over the gut microbiome, sir. And the kind of insights that have come through the talk about the gut microbiome have been really helpful. Slowly we'll also nourish our gut microbiome soon, the bioscience students. So I would request Professor Venkatesh, HOD Department of Biosciences, to hand over our love and token of appreciation to Professor D.N. Rao. It's such a joyous occasion that today happens to be the 70th birthday of Professor D.N. Rao.
Happy birthday to you. Happy birthday to you. Happy birthday, dear D.N. Rao sir. Happy birthday to you. May God bless you. May God bless you. May God bless you, sir. Happy birthday to you.
It's a very nice gesture on part of D.N. Rao sir that he chose to celebrate his birthday amongst us, sir. Thank you so much. Thank you so much, sir. Sairam, sir. Yes. I would like to request Dr. Balram Khamari, Assistant Professor, Department of Biosciences, SSSIHL Prasanthi Nilayam campus, to deliver the vote of thanks.
Sri Sai Ram, offering my humble pranams at Swami's Divine Lotus Feet. Respected Nagaraja sir, D.N. Rao sir, HOD, Bioscience Department, other department teachers, and my dear brothers, student brothers and sisters. Thank you so much for attending this program today, and it is my privilege to deliver the vote of thanks. I think this goes as for me, and as Swami had mentioned several times, this is the most important thing in any program, that we thank all the people who have made it possible. So to start off with, the unmentioned one, the hand behind everything, who holds us and who actually does everything, and we are just puppets. So I like to thank Swami for making this happen through all of us, this program. The second of them is our dear guest speakers who have come here today, taken their time out. And we know that sirs were not well physically, but they agreed to come here and talk to all of us today. Thank you so much, sirs, for coming here today. I would also like to thank the administration and the institution's Innovation Council for helping us collaborate with this and permitting us to have this program in our department. I would like to thank the director and the institute authorities for giving this auditorium to have this program today. And the campuses, the Anantapur campus and the teachers, thank you so much for coming here today, and having all of you is really a privilege for this program. Other departments who have come here today to attend this program, thank you so much for coming. The maintenance team who is working behind the scene to have all the PA system in order and all the screens to perfectly, especially the AVC department with Raghuram brother handling it. And all the faculty and students, especially head of the department, thank you so much. One more part is about the workshop which happened yesterday. We actually touched 65 students from 11th, 12th and undergraduate classes, and they all came to our labs. I think this is the first of a kind experience for them. from our schools. They had a hands-on experience. Every student was able to do gram staining by themselves. They streaked bacteria plates by themselves, and many other things they did on their own, which actually they never get to do as we were told by the teachers who came. So they were really happy, and it filled our hearts with so much of joy to see them happy. So also this whole thing was handled by the AMR laboratory. So I would like to thank the AMR, not this, something better. So thank you all the members of the AMR laboratory for carrying out this whole thing. I hope next time we will make this even more bigger, this International Microorganisms Day. And then this is a good start to start off with. And then thank you so much to everybody. And we will close this session with Arati. We request D.N. Rao sir to give the Arati to Swami. ओम जय जगदीश हरे स्वामी सत्य साई हरे भक्त जना संरक्षक पति महेश्वरा ओम जय जगदीश हरे शशिवदना श्रीकरा सर्व प्राणपते स्वामी सर्व प्राणपते आश्रित कल्पलतिका आपद्बान्धव ओम जय जगदीश हरे माता पिता गुरुदेवमु मरियंतयु नीवे स्वामी मरियंतयु नीवे नाद ब्रह्म जगन्नाथ नागेन्द्र शयन ओम जय जगदीश हरे ओंकार रूप ओजस्वी साई महादेवा सत्य साई महादेवा मंगल आरती अंदुको मंदर गिरिधारी ओम जय जगदीश हरे नारायण नारायण ओम सत्य नारायण 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 ओम नारायण नारायण ओम सत्य नारायण नारायण ओम सत्य नारायण नारायण ओम जय सद्गुरुदेवा नारायण नारायण ओम सत्य नारायण 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 ओम नारायण नारायण ओम सत्य नारायण नारायण ओम सत्य नारायण नारायण ओम जय सद्गुरुदेवा नारायण नारायण ओम सत्य नारायण 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 ओम नारायण नारायण ओम सत्य नारायण नारायण ओम सत्य नारायण नारायण ओम जय सद्गुरुदेवा ओम शांति 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 समस्त लोकाः सुखिनो भवन्तु समस्त लोकाः सुखिनो भवन्तु समस्त लोकाः सुखिनो भवन्तु ओम शांति 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 जय बोलो भगवान श्री सत्य साई बाबा जी की जय।
Sai Ram.